Hi, my name is Rafael. I'm a PhD candidate at University of British Columbia with Dr. Sally Aitken, and I study climate adaptation in Douglas-fir. But today I'm going to be talking a little bit about a very interesting approach to detect adaptive genetic variation associated with any particular trait. In this case here, we are applying this case-controlled GWAS to Douglas-fir and drought hardness. So my talk is divided into two main parts. The first one, I'm going to talk about drought hardness variation across the range of the species, so among provenances variation. And in the second part, where I'm specifically talking about the case control GWAS, exploring a little bit the within provenances variation in terms of drought hardness. So it's known for quite a long time that populations of conifers in temperate and boreal regions are locally adapted, and the main driver of local adaptation in this case is cold temperature. However, with climate change, droughts are becoming more frequent, more intense, and longer, increasing mortality and reducing productivity in forests. So we expect drought to become a very strong force of selection, and maybe driving local adaptation. Douglas-fir is a very important species in Western North America, ecologically and economically, but also in other parts of the world where it's planted. There is some evidence in the literature for local adaptation to drought in Douglas-fir. However, most of the studies were conducted with adult trees or saplings and focused basically on one specific region within the range of the species. So we are very interested to know how drought is distributed across the entire range of the species for the two varieties. And also we know that seedlings are strongly affected by selection to drought. So we were also interested to know how selection to drought in seedlings was distributed across the range of the species. So with that in mind, I asked two main questions here. How is drought hardness in seedlings distributed across Douglas-fir natural range? And also, how strong is the signal of local adaptation to drought in Douglas-fir seedlings? To try to answer those questions, I established a common garden experiment in a greenhouse with 87 seedlots or provenances. And those provenances represent a wide range of environments. In the experiment, I divided half of the plants that I submitted to an extreme drought condition, so drought to death, and the other half I used as a control to test for the difference. Every box that you're seeing here had at least one plant from each provenance. And here is the experiment. So I measured chlorophyll fluorescence across time, multiple times on each individual. So I used those chlorophyll fluorescence measurements in this simple linear regression approach, where I extracted the slope of those lines of those fitted lines as a proxy for drought hardness. And here we have two examples, one of a less hard individual and the other one with a more hard individual. So I partitioned the variance that I observed in this experiment, and I was able to observe some of the variation coming from between varieties. And this is pretty clear on the map here, interior and coastal being very different. So I tested for this difference and detected a strong difference between the two varieties. So I went a step further and separated the two varieties to partition the variances, and the pattern is pretty similar but not the same between the two varieties. So most of the variation is within provenances, and for the interior varieties, some signal of local adaptation was also detected, with 7% of the variation being explained by among provenances variation. So with with that in mind, I went a step further again and explored the within provenance variation to try to identify putative adaptive genetic variants associated with drought hardness in Douglas-fir. So here I'm exploring the within provenance variation. I conducted a second experiment with a subsample of the populations that I used in the first experiment. So for each provenance, I had 80 seedlings in one single box. So we can say that each provenance has had its own experiment. And so here we have, let's say, provenance 1. This is just one example. After phenotyping 6 times for chlorophyll fluorescence and making and extracting the slopes again, I could rank those individuals and look at this variation to select cases and controls within each provenance. So cases being least hardy individuals and controls being most hardy individuals within each provenance. So with those cases and controls, we went to each group and extracted the DNAs of those individuals and pooled them together in DNA pools. We conducted a sequencing using target exome capture, so a technique called pool-seq. After filtering and calling the SNPs, we were able to assign allele frequencies to each case and control within each population and test for the association between the difference in allele frequencies between these two groups and the phenotypes, so drought hardness. I used specifically for this test the CMH test. Here is the experiment over time. You can see that I really tortured the plants here. So I ranked the individuals in terms of the slope and separated the two groups, most hardy from least hardy individuals, and tested for the difference between these two groups. And for all the populations, I was able to separate individuals pretty well within each population, so large variation within provenance. And with those two groups, I used the allele frequencies from each group and tested with the CMH tests and was able to detect a large amount or a large number of SNPs for the coastal and the interior variety here. So with that, I conducted an enrichment test, and there are many goal terms that really stood out to me. And of course, I still have to explore a little bit further this year because it's very incipient result, but very interesting results. So with that, I would like to conclude by saying that signal of local adaptation to drought is weak for the interior variety, but is almost absent for the coastal varieties, again, in seedlings. A substantial amount of the variation within provenances in both varieties was observed, and we were able to utilize this variation within provenances to detect adaptive genetic variants associated with drought hardness. So with that, I would like to say thank you very much to all the collaborators and people who helped out with the experiments, and also people who provided seeds. Thank you very much.
My name is Susan Mcevoy, and I'm a PhD student in the Plant Computational Genomics Lab at the University of Connecticut. Today I'll be talking about 2 different maple species and their strategies for responding to abiotic stress. So first we'll go over a little background information, then the 2 different methods that were integrated to compare the maples with very different strategies that were reflected at the genomic level. The 2 maples we're comparing are Saccharum or sugar maple and Negundo box elder. So starting off with trait differences, Saccharum is very long-lived, 400 years on average, Negundo about 60 years. Saccharum's reproductive age is 40, the other is 5. One is monoecious, the other dioecious. Saccharum has high nutrient demands and is sensitive to deficiencies like calcium and magnesium, and it is more sensitive to salt and pollutants. So Negundo's native range is very broad, shown here on the map in orange. Saccharum is blue and is much more limited. We're interested in comparing these trees for issues related to each. Saccharum is known for maple decline. This is generally crown dieback, shown here in the photo, and an overall decrease in health and vigor. The dieback provides opportunities for species more tolerant of acidic soils, resulting in shifts in forest composition in its already limited range. The decline is thought to be in part a result of the higher nutrient demands mentioned on the previous slide, in particular calcium deficiency. Ngundo, on the other hand, is considered invasive in parts of Europe, South Africa, and Asia. So this is largely attributed to its plasticity in both overall growth and allocation to biomass, though it was found to have lower rates of photosynthesis and nitrogen content compared to natives, which is interesting because disadvantageous traits are not normally found in invasives. It also has phenotypic differences in sex expression, so females are more successful in wet riparian habitats, males in dry due to better stomatal regulation, and they have differences in growth. For comparison, I made two reference genomes and annotations. Starting with very high coverage Illumina and PacBio sequencing. PacBio was used for the draft assembly in Falcon, followed by Hi-C reads for scaffolding. Saccharum is 626 megabase pairs with an N50 of 45 megabases. It's very complete with most of the sequence organized into 13 pseudochromosomes shown here in the Hi-C mapping image. Ngundo is a little smaller at 442 megabase pairs and an N50 of 32 megabases. It's also in 13 pseudochromosomes, and both had high Busco scores, which is a measure of completeness using highly conserved single copy orthologs. So Sacharum's score has slightly more duplication. And after annotation, Saccharum had 35,000 genes and Ngundo had 27,000. So next we'll move on to the methods. So the first set of data we generated was from comparative genomics. This was done including Acer yangbiense, the only other Acer genome available at the time, and about 20 other plant species. So you take all the genes and group them into families by sequence similarity, and this can be used to predict evolutionary changes in families. We can look at relative sizes across species, rapid expansion in the most recent branch, and rapid contraction. And by assigning functions to these gene families, we can determine enrichment across the whole genome and also focus on specific functions of interest. The second set of data was transcriptomic and only for Saccharum. It was from the Hubbard Brook Experimental Forest, a long-term ecological research station in New Hampshire. They're running a long-term nutrient perturbation study, including plots that have been amended with calcium and others with aluminum. The interaction is shown here on the tree. So increased acid deposition in the soil causes leaching of cations like calcium and magnesium. It also causes mobilization of aluminum and conversion to more toxic forms. Members of the lab extracted RNA-seq from stem tissue. They were collected from 3 trees from each of the plots, calcium, aluminum, and control. And this was done in fall and again in spring. And I used the RNA-seq and the reference genome and annotation to do a differential expression analysis. So moving on to the results, we can start to see how these species differ. The overall picture is that Ngundo is a much more contracted genome compared to Saccharum. So this is a circus plot with Saccharum chromosomes labeled in blue and Ngundo in orange. You can see Saccharum is larger, taking up a larger portion of the circle, and it has more transposable elements shown in purple. The gray lines show synteny between the two. While Ngundo shares a high level of macrosynteny with yangbiense, we see here what appears to be a large-scale translocation in Saccharum. That's the darker gray and black lines. So during the whole genome duplication analysis, which involved separating out different kinds of duplication, it was shown that Saccharum had about a third more duplication in total. The last two figures are overviews of the genomic comparison. This first Venn just shows the scale and amount of overlap in the resulting number of gene families. The last one is the total of rapidly expanding or contracting families for each species. Saccharum has many more expanding, while Ngundo really didn't have much and was mostly contracting families. So the overall picture is that Ngundo is much more reduced. If we take the gene families that are larger in one Acer relative to the others, and we focus on gene ontology terms related to stress, you can see how Ngundo is fairly limited compared to Saccharum. Ngundo has cadmium, drug, chemical, bacteria, and UV, but Saccharum It's a lot more related to water, temperature, and a broader assortment overall. So now I will show just a couple of examples from the differential expression analysis. This is from spring, which had a much larger set of differentially expressed genes overall. There was so much associated with stress, but not much really specifically associated with aluminum response. There are a couple at the top highlighted with bright pink borders. And now this figure shows the relative gene family sizes for some generally associated to aluminum, but not specifically with the expression data. The darker colors are larger families, Negano and Saccharum on the left. So you can see of the blue set, there aren't many that are larger. Metallothionein like 3 is, and that is in our expression example, as is aluminum activated malate transporter, which is a smaller gene family. The oxidation reduction families are in purple, and the list is mostly ones that are seen in the expression data, and they do tend to be larger for Saccharum. Gene family size doesn't necessarily correlate to expression values, so it's hard to say why we don't see expression of more of those aluminum-related genes, and it could be a number of factors. It could be that it is stem tissue, or that these trees are more acclimated versus testing a more immediate response. The second example is from fall. There weren't any aluminum response genes here, but there was an interesting expression of accelerated cell death 6 and related genes in calcium plots. This gene participates in natural and stress-related senescence and helps with nutrient remobilization. Our version of ACD6 is a variant compared to the Arabidopsis model, and it belongs to a small gene family specific to the sapindales, which seems to have greater variation in protein domain copies and placement. This seems to be a good example of Sacharum's high calcium requirement to facilitate normal processes. Various types of calcium signaling related genes were seen in both the expression data, and it was an area of some expansion in the gene families. The last example is from Negundo. The fact that it is generally contracted makes the areas where it is expanded more interesting. There was enrichment in DNA damage and repair functionality, which could be further categorized in subgroups related to meiosis, UV and translesion synthesis, developmental processes, and abiotic stress. So here is where we actually see more response than Saccharum, which makes me wonder if Negundo's response, or its plasticity and growth in optimal conditions versus mere survival in other, hinges more at the DNA damage level than these very specific abiotic stress responses seen in Saccharum. But there is a lot more work to do, and we would need expression data for Negundo and additional studies for Saccharum to say more. The next steps for these particular Acer species is to look at what role methylation could be playing in these response mechanisms. I have nanopore sequencing for both that will be used for detection of modifications and creation of methylation profiles for both Saccharum and Negundo. With that, I'd like to thank my advisor, Dr. Jill Wegerson, Uze Sezin, Alex Trend, and members of the lab, and thank you all for attending my presentation.
Hi everyone, this is Mengjun. I got my PhD degree in Dr. Emily Moran's lab at UC Merced last summer. After that, I worked as a postdoc researcher in the same lab. My talk today is about association genetics of drought tolerance in ponderosa pine. The left figure shows the California drought between 2013 to 2016, which was the most severe drought in the past century, leading to over 100 million standing dead trees, showing on the right side. Given the predicted temperature increase in the near future, this kind of hot drought may represent an increasingly common situation in California. Understanding adaptive genetic variation is critical for local species persistence under changing climate, with limited studies going on in non-model species. In this study, I chose ponderosa pine people as a focal species. People is an ecologically and economically important species. It tolerates a wide range of temperatures and precipitation. It is regarded as one of the most drought-tolerant trees in North America. Genetic resources are fairly well developed in conifers, with available methods of sequencing and data analysis, as well as available reference genome of loblolly pine. Despite the importance of people, no previous study has investigated the genetic basis of drought tolerance in this species. We use two types of genetic association methods in this study. G2E, genotype to environment association, can be useful tools in the search for genetic loci responsible for local adaptation. However, the physiological traits that may be associated to the loci is unknown. In G2P, genotype to phenotype association analysis, the phenotypic data need to be measured in a common garden environment in order to eliminate the effects of environment. It can be useful tools in identifying genetic loci linked to targeted traits. However, whether these genetic loci are under selection in nature is unknown. Thus, G2E and G2P are complementary. In this study, we combine G2E with G2P to address the knowledge gap about genetic basis of adaptation to different climate conditions in people. So how to get the genetic data? In the 1970s, the Forest Services planted clones of over 300 wild people in Chico Orchard. The location is shown at the star place on the right figure. These people came from diverse climate conditions in the central portion of California's Sierra Nevada mountains and are now reproductively mature, which presents an excellent resource for genetic studies. For this study, we choose 223 individual people from the Orchard connection with the source location shown as the black dots in the right figure. Fresh needles were collected and dried with silica gel desiccant. We then extracted the total genomic DNA from the dried needles. We use GWAS, genotyping by sequencing, to sequence DNA, which is a restriction site-associated DNA sequencing technique and can produce large amount of SNPs in a relatively cheap way. Conifers has large and repetitive genomes. For the first step, digest DNA with restriction enzyme. If we use the methylation-sensitive restriction enzyme in step 1, we can exclude repetitive regions by avoiding cutting the most methylated repeated DNA sequences. Second, each sequence will be ligated to a forward adapter, a unique DNA barcode on the one side, and a reverse adapter on the other side. We can use a barcode to assign sequences to samples. Then we pull the DNAs, ran the PCR amplification, and got the sequence data. So for the 223 people, we ran GWAS with APKI as restriction enzyme to get the sequence data. We ran STAX reference-based pipeline with the genome of lobolipine as a reference genome to call SNPs. We identify over 4 million SNPs in total. However, many SNPs fall in the intergenic regions and have no direct effect on gene expression or function. So we filter out these intergenic SNPs, keeping about 900,000 SNPs in or near genes for the association analysis. I chose LFMM, the latent factor mix model, to run the association analysis. The number of population K represents the latent factor in this model. We estimated at the mixture coefficient for each K value between 1 to 6 with 10 repetitions. The figure on the right side shows the results. The most likely K value was identified by minimizing the cross entropy value. Accordingly, K equals to 1 is the best choice. To get the environmental data for G2E analysis, I extracted a 30-year average climate data between 1921 to 1950 from California BCM. PCA analyses were conducted among all the 18 climatic variables, and we chose 5 of them, which represent different directions of PC with low to moderate correlations, including maximum temperature of summer, minimum temperature of winter, precipitation of winter, climatic water deficit, and April 1st snowpack. Here is the result of G2E running with LFMM. The table on the left side shows the number of SNPs associated with the 5 chosen climatic variables, as well as the location of SNPs in genes. PCK4 April 1st snowpack had the most association, with TMIN, the minimum temperature of winter, has the next highest number of associations. The figure on the right side shows overlapping SNPs among these climatic variables, while Tmax, the maximum temperature of summer, has no overlapping SNP with other variables. The number of SNPs associated with more than one climatic variable was low, with the highest degree of overlap between PCK4 and TMIN 64 SNPs, and between CWD and TMIN 17 SNPs. For the greenhouse experiment in G2P, we used PVC pipes to build 10 boxes. We put 100 plastic tubes in each box with 1,000 plastic tubes in total. The plastic tube is about 120 centimeters depth because the maximum root length in a pilot experiment was more than 110 centimeters. 5 boxes were assigned as wet treatment and the rest 5 boxes at dry treatment. We collect seeds from 48 previously genotyped mother tree. Potentially, we have 20 seedlings for each family, with 10 in dry treatment and 10 in wet treatment. After the germination and growth for about 2 months, the wet treatment group was watered twice every week, and the drought treatment was watered once every 3 weeks for about 4 months. After obtaining the physiological data, we ran ANOVA in R to test drought response trees by comparing the data from wet and dry treatment while accounting for block effects. As shown in this table, 6 out of the 8 measured trees were significantly different in the drought treatment versus the wet treatment, including root length, shoot weight, root shoot ratio, stomata density on daxil side, number of stomata row on baxil side, and height growth. For G2P, we use the average trace value in wet treatment at the breeding value of mother tree to run the LFMM. The table on the left side shows the number of SNPs associated with the 6 drought response variables, as well as the location of SNP in genes. Root to shoot ratio had the most associations, while the shoot weight has the least associations. The number of SNPs associated with more than one trait was also low in G2P analysis. The highest degree of overlap was root length and root shoot ratio with 12 SNPs, and between root shoot ratio and the number of rows in a baxil site, 9 SNPs. There is no individual SNPs overlap between G2E and G2P, but the associated SNPs did share similar gene function categories, including the ubiquitin, seed pollen and ovule formulation, cell wall formation, stress response such as F6 exit, and cell division and growth. Any questions? Thank you.
Hello everyone, my name is Depardieu. I'm a postdoctoral researcher at University Laval under the supervision of Jean Bousquet and Nathalie Isabelle. Today, I will talk about how we can integrate tree-ring phenotypes, genetic associations, and transcriptomics to decipher the genomic architecture of drought adaptation in white spruce. Of course, despite climate change, we want to preserve a healthy and productive forest. Insect attacks, fires, as well as extreme climatic events such as droughts have a major impact on forests. Faced with this problem, several strategies can be adopted. In the case of drought, it is necessary to plant trees that are tolerant to water shortage. If we work with a single species, we can exploit the genetic variability naturally present in these species to identify and replant trees that are resilient to future environmental stresses. Climate change is extremely rapid, while the evolution of species is relatively slow. This is particularly the case for conifers, which are long-lived and slow-growing species. At present, we need to better understand the physiological mechanisms involved in tree adaptation to climate change. Specifically, we need to identify species, populations, and even stress-resilient trees. Until now, the genetic basis of drought tolerance remains poorly documented in conifers. That's why we need to clarify the relationships between plasticity, here I mean the response of a functional trait to a single stress event, and genetic variation. We also need to determine which traits and genes are linked to drought tolerance. Now let's define the concept of resilience. Imagine a person who is passionate about her work and very productive like me, until that person experiences stress and decreases her productivity. We define recovery as the ability of this person to re-increase her productivity after the stress period, while resilience is defined as the ratio of productivity before versus after the stress. I bring you here to Quebec. Here, our study site is indicated by a red star. We studied a total of 1,481 trees from 43 populations located in relatively dry areas, shown here in orange or red, or in relatively humid and blue areas. Our trees were phenotyped and also genotyped for thousands of candidate genes. This graph shows the growth versus time for the 43 provenances studied. As you can see here, a major drought caused a significant loss of growth for all provenances in 2001 and 2002. We then examined resilience as a function of long-term growth. We observed that populations from dry geographical origins grew better and had higher resilience to drought, compared with populations from more humid geographical origins. Interestingly, there was a lack of trade-off between resilience to drought and growth. We also estimated the narrow sense irritabilities for 3 resilience indices and found out that growth resilience was the most irritable trait in our study. Taking together, our results suggest that we could at least maintain drought resilience under future climate context. Now let's move on to our second objective. In the paper we recently published in Molecular Ecology, we wanted to determine which traits and genes were related to drought tolerance in our study species. To examine the genomic architecture underlying adaptive variation related to drought tolerance, we performed genotype environment association or GEA analysis and genotype phenotype associations, also called GPA analysis. We considered both conventional and tree-ring traits in our GPA analysis. We identified adaptive genetic variation by correlating allele frequencies for candidate single nucleotide polymorphisms with temperature, precipitation, and aridity gradients using 2 different methods. For GPA analysis, significant associations were tested between genotypes and 11 conventional and tree-ring traits using a polygenic model. Conventional traits included height and diameter at 38 years old. The first category of tree-ring traits were represented by some tracheic features such as cell wall thickness, wood density, and radial lumen diameter. Growth resilience, recovery, and relative resilience in response to the major drought event represented the second category of tree-ring traits. The climate sensitivity category captures the overall effect of drought on annual fluctuations in tree wood density, cell wall thickness, and lumen diameter across tree lifespan. After genetic associations, we added transcriptomic data from an accompanying growth chamber experiment to investigate differential expression under drought conditions. 285 genes were significantly associated with a climatic factor or a phenotypic trait, including 110 that were differentially expressed in response to drought. To conclude, we showed that for white spruce, adaptive genetic variation in response to changing local conditions can shape drought vulnerability. Have we found the magic recipe yet? Certainly not. It would be relevant to confirm the patterns identified here by integrating data from other common gardens. Since tolerance to water stress may involve many phenotypic traits, we are currently analyzing the interannual variations of carbon-13 and oxygen-18, as well as variations in wood anatomy during the growing season. I wish to thank the organizing and scientific committees of the CFGA, all my colleagues at Natural Resources Canada and Université Laval, our collaborators and our funders. At the end of this session, I will be available to answer a question live. Thank you for your attention.